Assalamualaikum warahmatullahi wabarakatuh to all my students, the U1 Block 4 students. Alhamdulillah, today we will continue with the second lecture out of 4 lectures in regard to gastrointestinal tract system. I believe that this is our first encounter each other since I just newly reported duty to Kuliah of Medicine in Physiology unit. So we better start with a little bit of introduction or ta'aruf. So my name is Dr. Nur Baiyah binti Muhammad Bakrim. I graduated with MBBS from Monash University, Australia in 2008, and I worked as a medical officer back in Australia for 3 years and came back to Malaysia in 2012 and then joined UIA as a trainee lecturer. So I proceeded with a master and also PhD, and Alhamdulillah just completed my PhD in physiology in recent June 2020. That's why this is my first lecture with all of you. I hope that you bear in mind that I'm learning as well, especially because this is the ERTL, the online teaching platform that we are using. All of us are trying to adapt with this new norm. Inshallah, we are trying our best. Okay, so the topic that we are going to cover today is still related to upper gastrointestinal tract, part 2, but I'm focusing on secretory and motility function of stomach. So in clinical practice, when you are talking about gastrointestinal tract system, it is divided into 2. The first one is upper GI and the lower one is and the second one is lower GI. Upper GI involve gastrointestinal tract started from mouth, esophagus down to stomach, while lower GI is referring to duodenum, which is the first part of small intestine, and large intestine and rectum. So at the end of this lecture, I hope that all students will be able to state the general function of GIT system, and secondly, able to state the general effects of parasympathetic and sympathetic neural activity, and also state the site of release, stimuli of secretion, and actions of GIT hormones. So basically the first part of this lecture, just to revise a little bit about introduction of GIT system. In general, the gastrointestinal tract that actually start from your mouth down to your rectum involved in 4 main functions. The first one is digestion, which is referring to the breakdown of the complex form of food that we eat that in the form of fat, protein, and carbohydrate. They need to be broken down into a simpler form of nutrition to be absorbed in the small intestine. The second function is motility, which is referring to the movement of the food in terms of bolus via muscular contraction down to the stomach and then to the small intestine, large intestine, and then to be excreted via rectum. So motility referring to the movement of the food itself. Another function of gastrointestinal tract is for secretion, either exocrine, meaning that secretion of enzymes and also digestive juice, for example hydrochloric acids, to actually digest the food, as well as secretion of endocrine, meaning hormones. The difference between exocrine secretion and endocrine secretion is exocrine meaning that enzymes and this digestive juice is secreted by cells direct to the stomach, to the GIT tract. Meanwhile, for endocrine, hormones is actually secreted to the bloodstream and then they act to the particular organ like stomach or small intestine via blood vessels. And the fourth function of GIT tract is for absorption of nutrients, primarily via small intestine. So we will look into more details later on in this lecture. In terms of nerve supply of gastrointestinal tract, basically in normal routine, if you are happy, relaxed, conditioned, the baseline of nerve supply is actually our GIT tract were supplied by 2 systems, extrinsic innervation, which means that the nerve is originated from outside of digestive tract, and secondly, intrinsic innervation, in which the nerve is actually derived from within the digestive tract. And if you are in normal condition, our GIT tract is actually innervated by parasympathetic reaction, meaning that's why if you are in normal condition, normally you can eat with normal appetite because there is an increased intestinal smooth muscle activity, also stimulates motor and sensory secretory activity. However, if you are in upset, there is an upset event, or you are in a stressful condition, you normally will result in loss of appetite because there is in sympathetic reaction, there is a decrease of intestinal smooth muscle activity and sphincter and blood vessel contraction, meaning that you don't feel like eating when you are actually in sympathetic reaction. Meanwhile, for enteric nervous system, intrinsic innervation, which also known as enteric nervous system, it is divided into mesenteric plexus and also submucosal plexus, which actually control the motility of the food, specifically control of peristaltic activity, and also for submucosal plexus, it control the secretion of exocrine and also endocrine. This table shows us the different types of GRT hormones: gastrin, CCK, secretin, and also GIP, gastric inhibitory polypeptide. I'm not going through into details because you will go through each hormones as we go on. So for the second part of this lecture, we'll have a look into secretory function of stomach. At the end of this lecture, I hope that students will be able to state the anatomical regions of stomach and also describe the functions of stomach, state the constituents of composition of gastric secretion, and describe mechanism of gastric acid secretion and its regulation. So you can imagine this is your stomach, in which we already covered esophagus, and once the food actually arrives, opening of stomach, which is known as cardia region, the food is in the form of bolus. And then if there is already small part of there is already food in the body of stomach, then the food will be stored in the fundus area, which is also known as a reservoir. And this is the main site of food digestion, and gastric juice secretion mainly taking place at the body of the stomach. And the food will be moved forward to the pylorus region, in which it is being prepared to be passed to the duodenum, which the process is known as gastric emptying. And here the gastrin hormone is secreted, and the food will be ground into smaller particles to be passed into the duodenum region. And if you are looking into details, we take one part, small part of the stomach wall. Stomach wall is actually embedded with different types of cells, which actually secrete different types of enzymes and also gastric juice. If you're looking into here, there is a chief cells that is responsible for pepsinogen secretion. And then we have these parietal cells, in which acid hydrochloric actually been secreted from parietal cells. And there is also mucous neck cell, which actually secrete mucin, which have the protective effect or function to the layer of the stomach against the acidic environment as a result of hydrochloric acid secretion. So what is the main function of hydrochloric acid? It is actually to convert pepsinogen to active form of pepsinogen, which is known as pepsin. And this is the enzymes that actually will digest the protein into their simpler form, which is peptides. If you're looking back into the previous slide, okay, we're moving forward. So with that structure, it will give us idea about the function of stomach. Stomach have a motor function, in which as a storage, as there is a fundus area of the stomach to store the excessive food, and also formation of chyme. So at the beginning, the food that arrives the stomach will come in the form of bolus, but at the end of food digestion in the stomach, the food will be converted in a chyme form, in which there is more fluid as compared to the bolus form, is more solid. And then there is also the emptying of stomach that actually has a role to passing the stomach content into duodenum. At the same time, stomach also has a secretory functions in terms of stomach lining secrete hydrochloric acid and pepsin, which involve in protein digestion. It also secrete intrinsic factor, which actually very important for vitamin B12 absorption later on in small intestine. And it also secrete mucin, in which actually have a mucus protection. We know that stomach is very acidic. That's why without mucin, the stomach lining will result in small severe damage due to the hydrochloric acid. But it's been protected by the mucin. And in pylorus region, there is also G cells that actually secrete gastrin, which is actually responsible to stimulate the gastric secretion and also gastric motility. And in stomach, there is also a small amount of lipase, which actually targeting the fat to be digested. The third function of stomach is protective functions, because if you're eating some food that contain bacteria or even poison, it's actually your stomach, the acidic environment of stomach will kill the bacteria within 15 minutes. And at the same time, if you're taking something that not good for your body, like a poisonous food, then your stomach will actually excrete or eliminate the food by vomiting mechanism. When you're talking about gastric secretion, there are 3 phases that involve in to secrete the gastric juice. When we're talking about gastric juice, referring to the fluid that been secreted in the stomach. In a day, we actually secrete about 2,500 ml gastric juice per day, and the pH is acidic, which is 1.0. Apart from this, there is also organic content like enzymes such as pepsin, lipase, and also mucus and intrinsic factor. At the same time, there is also inorganic content, for example, like cations, magnesium, hydrogen ions, and also anions like chloride ions. So during this, we have cephalic phase, gastric phase, and intestinal phase. During cephalic phase, meaning that the gastric secretion actually has been initiated even before the food entered the stomach. Stimulus such as like the smell of the food, and also even though when we chew the food in our mouth, and even though the food just arrived the esophagus part when we are swallowing, but then the gastric juice already been secreted. This is actually controlled by 2 stimuli, either unconditioned stimuli, which is referring to the impulse that derive from the presence of the food in the stomach. When you eat your food, for example, nasi lemak, and you put into your mouth, then the presence of the stomach in the mouth, actually there is a message or impulse sent to your brain, and then the brain accept the message, then due the vagus nerve, they will send the message back to your GIT tract, which is your stomach, to secrete the gastric secretion. And it is also controlled by the conditioned stimuli, meaning that impulse from cerebral cortex, just by looking into the food or the smell of the food, even the sounds of you opening the plastic wrap of the food, actually all those actually stimulus or message which is transmitted through the dorsal motor nuclei of the vagus nerve, and then they will send the message to the your gastric area, and then produce the gastric secretion. So during cephalic phase, even before the food arrived the gastric region, which is the cardia region of your stomach, about 30% of acid or gastric secretion already been produced. Once the food actually enter the stomach, there is more gastric juice will be secreted, and this is known as gastric phase. It is controlled by neural and also hormonal pathway. Basically, the bolus arrival causes distension of the body of stomach. So the more food that you eat, so your body of stomach will expand, and it activate the tension sensors. And these tension sensors will activate the vagal nerve to be activated, and this reaction we call as vagal reflex. Vagal referring to the stomach, vagal referring to the vagus nerve, reflex is the reaction of outcome. And then it stimulate parietal cells to secrete hydrochloric acid, and also stimulate chief cells to secrete intrinsic factors. That is how the gastric phase is controlled by the nerve. Secondly, gastric phase also controlled by the presence of hormones, particularly gastrin hormone, which is released by G cells at the pyloric region and in the mucosa of duodenum as well. So this gastrin hormone actually stimulates and promotes gastric secretion and gastric motility to optimize the digestion process in the stomach. And then gastric action only been inhibited as the pH of the stomach fall below 2.5, meaning when there is excessive or enough hydrochloric acid being secreted into the stomach, then the gastric action will be inhibited. The third phase, the intestinal phase, is more like inhibition phase, in which gastric secretion is inhibited so that the passage of chyme from the stomach does not overload the duodenum. So the process of digestion can be slowed down because there, at one time, the amount of food that been passed from or been emptied from stomach into duodenum cannot be too much, because the process in the duodenum take longer time and they need to digest to further break down the food and to absorb the food in very slow, take quite a long time. That's why it need to be bit and pieces a little and little, but then continuously. It involves negative feedback, which means that distension of duodenal wall is not the body of the stomach, but now is the distension of duodenal wall, and also the hyperacidicity, meaning that the food that coming from the stomach actually in acidic form or is acidic, and it will cause the acidicity in the duodenal area. And the presence of fats and peptides will activate the neural pathway to cease the gastric secretion. In simpler form, when there is enough food in the duodenal region, then there will be a negative feedback signal that telling the stomach that, okay, you can stop the gastric secretion, slow down, because we need to process this food first. And then intestinal phase also being controlled by hormonal action, which known as androgesterons, meaning hormone that's secreted by the small intestine, which are CCK and also secretin. And these two hormones will inhibit the gastric secretion, as compared to gastrin, which is that hormone will stimulate the gastric secretion. But for CCK and secretin, they have the opposite effect, in which they will inhibit the gastric secretion. So I hope everyone will understand the three phases of gastric secretion by now. So we'll proceed with the mechanism of hydrochloric secretion. It is secreted by parietal cells, which actually rich with enzyme carbonic anhydrase, which will be catalyze the hydration of carbon dioxide, meaning that carbon dioxide will combine with water, which is H2O. Okay, carbon dioxide combined with water, and then will form carbonic acid. In the parietal cell, carbonic acid spontaneously dissociates into hydrogen ions and also bicarbonate ions. So this is the formula. Carbonic acid dissociate into hydrogen ion and bicarbonate ion. And this diagram will show with a clearer picture, like carbon dioxide and also hydrogen and water, H2O, will combine, catalyzed by the presence of carbonic anhydrase, this is an enzyme, and carbonic acid to form carbonic acid. And this carbonic acid will spontaneously dissociate into bicarbonate ion, which is have negative charge, and also hydrogen ion, which actually present in positive charge. And this hydrogen ion will be actively transported out of the parietal cell into the lumen of the stomach. Lumen of the stomach referring to the hollow part of the stomach. Okay. Why is this actively? Because it's against the hydrogen ion gradients, meaning that hydrogen ion is actually more, the presence is more in this area as compared to this area, but through the active transport, they can be transported out of the cell. At the same time, just to balance the positive charge, as you take out the hydrogen ion, the potassium ion will be transported into the cell. Okay. At the same time, the negative charge of carbonic ion, the hydrogen bicarbonate ion, this is the bicarbonate ion, will be transported out into the bloodstream. Here, this is known as the anion exchanger channel, And once you take out the bicarbonate ion which is negative charge, it will be replaced by the chloride ions, will be transported into the cells. And then as the chloride ions concentration is higher in the parietal cells, it will be passively transported as per follow the gradients. This is higher gradient, higher concentration of chloride ions, and it will be passively transported out the cell into the lumen, and the opposite charge of hydrogen ion and also chloride ion will attract each other. Okay, so this is the more description. Hydrogen ion is actively transported into the stomach lumen by hydrogen and potassium ATPase proton pump. This is an active transport that need energy. This channel use ATP energy to exchange potassium with hydrogen in the parietal cell. And the bicarbonate ion is transported out of the cells into blood via the transporter protein called as an ion exchanger in exchange of chloride ion. And the chloride ion is transported into the stomach lumen via the chloride channel. And the opposite charge of hydrogen ion, which is positive charge, and chloride ion, which is negative charge, will attract each other, and they will combine later on, forming the hydrochloric acid. So there are a few factors that affecting gastric secretion. Firstly, hypoglycemia, meaning that if there is a low sugar level in blood, then there will be signal that being sent to the brain so that it can promote acid and pepsin secretion in the stomach to actually optimize the food digestion so that more glucose can be absorbed into the blood system. And then alcohol and caffeine also will stimulate mucosa to increase gastric secretion. That's why excessive alcohol and caffeine intake will actually cause gastritis or inflammation of the gastric lining due to the excessive gastric secretion. Histamine also can increase the presence of histamine, or this is inflammatory markers. If you get injured or you are in stressful condition, normally your histamine level will increase, and then it will act via hydrogen ion receptors on parietal cells and then resulting in increased acid secretion. That's why if you are in chronic stress, it can cause gastritis as well. And in stressful condition, emotional factors modify gastric secretion and circadian variation, meaning that in the morning, for example, or in the early evening, there is a higher acid output as compared to the late night and also in the afternoon. That's why we are encouraged to eat our dinner early night, because during that time, naturally, your acid output is higher, and if you are eating at late night, then you need to work harder to secrete the acid for food digestion. Okay, so we are coming to the third part of this lecture, motility function of stomach. At the end of this lecture, I hope that students should be able to state the different types of gastric motility and their importance, also to describe the control of gastric emptying, describe the role of hormone in gastric emptying, and describe the clinical implications of secretory and motility function of stomach. So we'll start with the regulation of GIT motility. Basically, the movement of food in the stomach is controlled by three ways. First one, it reflex that originated outside GIT, which is autonomic nervous system, long reflex, and secondly, by reflex originated inside the GIT, which is referring to the enteric nervous system, and thirdly is by the hormone or peptides or enzyme that are present in the stomach. There are two types of motility, meaning the food being moved along the stomach by two types of motility. If in the esophagus, peristaltic movement is the main mechanism of way to move the food down the esophagus. Meanwhile, in stomach, segmentation is actually the main way or methods to move the food along the gastric region. So the different Okay, so this is a difference, as the peristaltic movement will result in the foot to move forward. You know, when you contract, and then the muscle will relax and the foot will move forward down the gastric region. However, for segmentation, is the main aim or main objective is to break down the food, so that there is more surface of the food in contact with the gastric secretion to optimize the digestion process. And these two motility actually happen at the same time, meaning that while the stomach actually being contracted using the peristaltic wave, at the same time there is also segmentation motility that taking place. And now we are moving to the gastric emptying. It's actually referring to a process in which the stomach empties into duodenum. And this is accomplished by three mechanisms. First is the peristaltic contraction. Even though segmentation is the main motility approach in the stomach, but if you want to pass the stomach content into duodenum, you need the peristaltic contraction, because this is the contractions that can result in moving forward or pushing forward the foot into the pyloric valve and then to the duodenum area. And then systolic contraction of the antrum. As there is a contraction of the antrum here, the food that with more liquid will be pushed forward into the duodenum, and the food that more solid will be pushed back so that it can be digested further prior to be passed into the duodenum. And also reduction in size of stomach to increase pressure in the stomach so that the food will be easily pushed forward to the duodenum. So the control of gastric emptying, there are 5 factors that actually influence or give or affect the control of gastric emptying. First, the physical nature of the food itself. So the liquid will empty faster than solid. I think it is easily been understood. And food in the form of carbohydrate will be emptied faster as compared to protein, in which in turn will empty faster than fats. Okay. Meanwhile, for volume effect, the more food that you eat, it will cause distension of stomach body, and the distension of stomach body will increase the activity of gastroduodenal pump. This is actually, the gastro is the stomach, duodenal is the duodenum, meaning that the operation of passing or emptying the stomach content into duodenum is controlled by gastroduodenal pump, and the rate of gastric emptying via vagal reflex. So vagus nerve also control the rate of gastric emptying. In simpler word, the more food that you eat, then the rate of gastric emptying will be higher, as there is more distension of stomach body. And then the effect of duodenum, the presence of fat, product of protein digestion, and that acidic environment of the duodenum will actually decrease gastric motility. In other words, that when the duodenum become more acidic, more fat is actually entering, more food entering the duodenum, then they will give a signal to stomach to actually reduce the gastric motility. Fourth one is the role of vagus nerve. Stimulation of vagus nerve will increase the antral contractility and accelerate the emptying of solids, and also at the same time it relaxes the gastric fundus and slows emptying of liquids. And excitement fasten the gastric emptying, while fear, stress, or fight or flight response will actually slow down the gastric emptying. And fifthly, there is a role of hormones, in which secretin, CCKPZ will inhibit gastric emptying. Okay, this is the hormones that secreted in the small intestine. Yeah. So we reached to the final part of this lecture, which is the clinical implications. In simple form, I would like to highlight 5 clinical conditions that actually will reflect the importance of our lecture today, which is stress eating. What actually will happen when you are in stress? Will you eat more or will you eat less? So like our current lecture actually can provide us with a little bit knowledge or information how this physiology of gastrointestinal tract will affect when will be affected when we are in stress, and how actually the formation of peptic ulcer and also Helicobacter pylori infection in the stomach. Gastroenteritis also another clinical implication of the failure of the stomach to function as normal, and vomiting is a protective mechanism in order to protect our GIT tract to be, polluted by other foreign substance that will damage our body system. So however, in this lecture, I will not cover in detail, but we'll discuss this during our real-time interactive session on 4th of August, inshallah at 8 o'clock in the morning. So meanwhile, you can start Google or referring your textbooks or reading journal just to see how actually these clinical implications taking place in applying what today's lecture content. Okay, so inshallah that's the end of my today's lecture. I hope that you have good understanding, and of course we can optimize, we can actually use our real-time session in near future, in next week, to discuss further. If you have anything that you don't understand, you can start posting your questions so that we can actually answer during that session. Thank you very much. I'm sorry for any shortcomings. Hopefully we'll keep improving and learning from each other. Assalamualaikum warahmatullahi wabarakatuh.